Hey guys, welcome back. Today we're going to cover the reproductive system. This video will cover the first half of the chapter, which is kind of an introduction to the reproductive system, and then focusing on the male reproductive system. The female reproductive system will be covered in a different lecture. These are just the objectives. These are the things that you should be able to do by the end of the chapter. You guys could just use this to guide your studying. And continue, these are just the female reproductive objectives. The reproductive system is a little bit different than any of the other body systems for a few reasons. One is it's the only system that's not essential to life. We need all of our other body systems to live, right? You have to have a cardiovascular system. You have to have a skeletal system, an integumentary system, urinary system. All of those are necessary for life. The reproductive system, though, is not. It's not necessary for the life of the individual. And this is evidenced by the female reproductive system pretty much completely stopping function at menopause. Right? At menopause, the female stops going through that monthly ovarian cycle and monthly uterine cycle, and the woman continues to live just fine. There are some changes in physiology, but nothing that drastic. So the reproductive system is really not necessary for the life of the individual. This is also evidenced by children. Right? Children do not have a functioning reproductive system yet until puberty. And that brings us to our next point. The reproductive system is the only system that has a latent onset, meaning it begins functioning later in life. So when an infant is born, the reproductive system is not going through the normal cycles and normal functions that it will later in life. Right? We know that the full onset of the reproductive system happens at puberty, which is typically in like the early teenage years, sometimes a little bit younger than that for females. The reproductive organs in general are involved in the production of offspring, so making a baby, and they do this by creating germ cells called gametes. So the gametes would be like the ovum or egg in the female and the spermatocyte or sperm in the male. Obviously, the sperm and the egg then meet up and they produce a new being. The reproductive organs are also involved in the secretion of the hormones that maintain the normal reproductive system function. These are hormones, the sex hormones, right? Like androgens, testosterone, estrogens, progesterones. These are the sex hormones that keep the reproductive system functioning appropriately. These hormones also maintain the secondary sex characteristics These are the things that you can kind of see on a person that helps you figure out, hey, this is a male, or oh, this is a female. So in females, these hormones kind of guide the deposition of fat. So there's more fat placed in the breast area or in the hips and buttocks area. Whereas in males typically don't have that large deposition of fat in the breast area. Or in males, the deeper voice is guided by testosterone. The hair, right, the pattern of hair growth on the face, sometimes on the chest, that's all guided by the sex hormones. Male and female reproductive systems are different from each other. That's another really big difference between the reproductive system and the other body systems. Right, the cardiovascular system is the same in a man and a woman. Once you cut the body open and you're looking at the heart, you can't look at the heart and tell me if it's a male heart or a female heart. The other body systems are relatively the same in the male and the female, but the reproductive system is completely different. And that's both structurally, right? The organs and the structure of those organs is completely different in the male and female, and functionally. They do completely different things. In females, the female reproductive system we see produces 1 gamete per month. So 1 egg or ovum is produced in the woman per month. That's what happens at ovulation. The egg is released. Also, after that egg is released, if it gets fertilized, then the female reproductive system retains that embryo, which is then later a fetus, and it nurtures it until it grows into an infant. Also, the female reproductive system takes part in the birth of the infant and then nourishes the infant after birth, right, by producing milk or during lactation. So the female reproductive system has a really kind of wide scope of function. It produces that gamete. It holds on to the gamete if it, in fact, gets fertilized, nurtures it, allows it to grow during that 9, almost 10-month period, takes part in birth, and then also continues to nourish it for up to about a year. So that's a pretty intense functioning there. The male reproductive system is a lot less complicated. The male reproductive function is kind of quick and done. The male produces the sperm, and that's it. Once the sperm is ejaculated into the female reproductive tract, the male has no other part in reproduction. So he's not involved physiologically after sperm is released into the vagina. Also, males produce gametes in a completely different way than females. Females, we said, release 1 egg per month. Males, on the other hand, make millions of gametes all the time. So the male produces large quantities of gametes. Again, that's the sperm, every single day. Right? So 500 million sperm per day, and that's all the time, day in and day out. We will see later when we talk more about these that there are differences in the timing of the reproductive system function as well. We mentioned that for females at menopause, the reproductive system stops working. But that doesn't happen for males. The male reproductive system continues working much later in age. Sometimes testosterone levels will drop, but males are still producing sperm at 70, 75 years of age. Before we get into detail about the reproductive systems, we need to review the 2 types of cell division that occur in the body, because that's going to help us understand the way that the spermatocyte or the ovum or egg are produced, and it'll help us learn about the DNA that they have in them. So cell division is just one cell dividing into 2, right? This is how our body produces cells to grow or replenish old cells that are worn out. Cell division is how our body produces eggs and sperm. Now, there are 2 different types of cell division, mitosis and meiosis. And we need to kind of go over the differences between the 2. Mitosis, the first one you see here, this is the cell division that occurs in almost every cell in our body. So muscles, bones, blood cells, all these cells undergo mitosis. In mitosis, one cell divides to produce 2 cells. So there are 2 daughter cells that are produced. Okay, 2 daughter cells are produced. And those 2 daughter cells are exactly identical to the original parent cell. Right, so literally you just have like 1 cell making a copy. So 1 cell turns into 2 cells that are exactly the same. So all of our body cells have 46 chromosomes. Remember, chromosomes, that's your DNA. So when we look at the DNA that's in the cells, all of our body cells have 46 chromosomes total. We call those cells diploid. Di means 2, right? And when we look at these 46 chromosomes, there are 23 chromosomes, but we have 2 copies of each. So there's 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, right? All the way down to 23. So 23 times 2 is 46. So total, each of these cells that divides by mitosis is going to have 46 chromosomes. Again, this is normal. These are all of our normal body cells. Meiosis, right, so we'll say these are normal body cells. Meiosis is different. Meiosis is the cell division that occurs in our sex cells. So that means in the sperm and egg or ovum. So our sex cells have this special type of cell division that occurs. And in meiosis, meiosis involves 2 cell divisions. So it's not just one cell dividing into 2. There's 2 steps of that. So 1 divides into 2, and then each of those 2 divide into 2. So you have a total of 4 cells. The 2 stages that occur in meiosis are called meiosis 1 and meiosis 2. Again, what happens is that one original cell ends up giving us 4 gametes, or 4 cells. But each of these cells only has half the chromosomes of that original parent cell. So that original parent cell, we said, had 46 chromosomes, right? There was 2 of each of the 23. Well, now these gametes that we produce, the sperm or the egg, are only going to have half of that set. So they each only have 23 chromosomes. So 1 of each of the 23. We call that haploid. Think of it like half. So again, this means that they have 1 copy of each chromosome instead of 2 of each chromosome. The way that this, we'll talk about this in detail, but the way that this happens is you have your one parent cell, right? And it has 46 chromosomes. Before it divides, it copies all of its DNA. So it kind of, the first thing that happens is kind of like mitosis. It's kind of like that first cell division. So your one cell has 46 chromosomes. It copies its DNA, right? So it's got double that. And then the cell splits into 2 cells. Each of these still have 46 chromosomes, because remember, it copied it first. Now, each of these cells, these 2 cells that we just made, are going to divide, but they don't copy their DNA first. Okay, so they only have 46 chromosomes. They're not going to copy it, they're just going to split. So now we have 1, 2, 3, 4 cells, and each of them got half of that DNA. So each of these cells is going to have 23 chromosomes. So we end up with 4 cells with 23 chromosomes each. Now this is important because of the function of our sex cells, the function of the sperm and the egg. In any of our other body cells, if they only had half the DNA that they're supposed to have, they couldn't function, right? The DNA, again, it's the instructions for how to do everything in your body. Every cell needs a full copy of DNA, except for these 2 cells. And again, think about what the function of these 2 cells are. What does the egg and sperm do? They come together to make offspring, right? They come together to make a new baby, essentially. So if you have your sperm over here, and your sperm has 23 chromosomes, And then I have my egg over here, and my egg has 23 chromosomes. They meet up together, right? And they both bring 23 chromosomes to the table. So the new baby that they make, right, the new offspring that they make is going to have a total of 46 chromosomes. Okay? All right. Okay, so now that we've kind of got some background information on reproductive systems and cell division, now we'll get into the actual details about the male reproductive system and the female reproductive system. And we're going to start with the male reproductive system. Just a warning as we go through the chapter, there's a lot of anatomy involved here, just because the anatomy and physiology are so closely related that we will cover a lot of the anatomy as we go through. So starting with the male, two major organs of the male reproductive system are the testes. The testes are a pair of egg-shaped glands that hang inferior to the pelvic region, so hang inferior to the body, just posterior, just behind the base of the penis. The testes hang from the body in a skin-covered sac called the scrotum. So think about this. There's got to be a reason for this. Most of the reproductive organs and organs in general are in our abdomen or pelvis, right? They're enclosed in the abdomen or pelvis. It's really risky to have an organ hanging from the body with just a thin layer of skin and muscle surrounding it. So there's got to be a pretty important reason that we keep the testes out of the body like this. And there is. The reason is temperature. The testes have to be at a very consistent, very specific temperature in order for sperm production to occur appropriately. And that temperature is just a little bit lower than our body temperature. So if the testes were actually up in the pelvic cavity, they would be too warm and we wouldn't produce enough sperm. So we have to keep them outside of the body so that they're a little bit cooler. Now, we have something called the Cremaster Muscle that helps control the temperature of the testes. And it does this by controlling their position relative to the body. So underneath, like say this is the bottom of the pelvis. Underneath the bottom of the pelvis, you have like the scrotum hanging down like this, and it has the testes inside it. The Cremaster muscle circles around the testes like this, and it goes around something called the spermatic cord, and then it goes and it connects to the abdominal muscles. And when this Cremaster muscle contracts, right, you can picture this, if this muscle were to contract, it would raise the testes up closer to the body. That warms them. Then when it relaxes, it drops the testes down further from the body, and that cools them. So by contracting and relaxing, right, or raising and lowering the testes, the Cremaster muscle can control how warm or cool they are to make sure that the temperature is just right for spermatogenesis or sperm production. Inside each testis is both, testis is just one. Inside each testis, there are septa, which are just little like dividers that subdivide the testes up into smaller little areas that we call lobules. So each of the testes is divided up into little segments or little lobules. And each of the lobules contain hundreds of really tiny, tightly coiled tubules that are called seminiferous tubules. Seminiferous tubules are the place where sperm is actually produced. So sperm production, the initial production of these sperm, occurs inside these seminiferous tubules in the testes. That sperm production is called spermatogenesis. Genesis is like to give rise to something. right? Like creation of the earth in the Bible is supposed to happen in the book of Genesis. So Genesis is to give rise of something. So spermatogenesis is giving rise to the sperm, producing the sperm. Now, there are small little ducts that are going to connect these seminiferous tubules to a structure called the epididymis. The epididymis is this really tightly coiled duct that goes up along the posterior border of the testes. So like if this is one of the testes, there's this really tightly coiled tube that goes along the back of it like this. That's the epididymis. And the sperm will go here into the epididymis to mature. So they're produced first in the seminiferous tubules, then they go through, so the seminiferous tubules in here, and then they go through these little tiny ducts to go enter into the epididymis, which is where they'll mature. Okay, so here you can see all of this. First, this out here is the scrotum. Okay, so the skin-covered light sac that kind of goes around the outside of the testes, that's the scrotum. It has a muscle in it called the Dartose muscle. The Dartose muscle, again, it's part of the scrotum, part of this skin-covered sac. And when the Dartose muscle contracts, it wrinkles the skin of the scrotum. It wrinkles up the skin of the scrotum, and that actually thickens it. That thickens that skin, which helps to warm the testes inside. So you see the scrotum on the outside with the Dartose muscle. Underneath the scrotum, you can see the Cremaster muscle. So the Cremaster muscle is this kind of pinkish color that you see, this kind of darker pink color that you see around them. Again, that surrounds the testes, and then it's going to also connect to this spermatic cord that we have, like going up like this, and then that'll connect to the abdominal muscles. Inside, you see the actual testes. So like this is a testis, and then over here is the other testis. Looking inside, there are septa, right? A septum is just one septum, so like this is a septum, this is a septum. These septa are just these walls that divide the testes up into little lobules, little areas. And inside each of those little regions, we have the seminiferous tubules, tons of really tightly coiled little tubes where the sperm are initially produced. It does take a long time for the sperm to actually be ready to go fertilize an egg. They take a long time to mature. So the sperm are initially produced in the seminiferous tubules, but then we'll see like their journey is long. They spend a while in the epididymis, and then they spend time in the Ductus Deferens, and in the ampulla of the Ductus Deferens. Like there's a whole process, but it starts in the seminiferous tubules. Then we have multiple little tubes or little ducts that will carry the sperm from the seminiferous tubules to the epididymis. Epididymis, you can see the top of the epididymis right here. It does coil along like the back of the testes. There are straight tubules. So you'll notice this straight tubule right here, right here, right here. And then they go back to the ret testis. And then that brings the sperm back into the epididymis. So we start like here in the seminiferous tubules. Then we go to the straight tubule. Then the ret testis, then the epididymis. The seminiferous tubules that fill the testes are each surrounded by connective tissue capsules. And then the space, the little space that's in between each of the tubules is filled with areolar tissue, which areolar tissue, remember, is just a connective tissue. Blood vessels, there's blood vessels in there, they are vascular. and then large interstitial cells. These are called Leydig cells, but these large interstitial cells or Leydig cells are very important for sperm production because these cells produce the androgens. Androgens, remember, are the dominant male sex hormones. So androgens include hormones like testosterone or dihydrotestosterone. And this testosterone, we'll see, is going to be very important for sperm production. It's very important in stimulating sperm production. So sperm are produced in the seminiferous tubules. Surrounding the seminiferous tubules, we have Leydig cells. Leydig cells produce testosterone, which stimulates sperm production. We said spermatogenesis is sperm production. So spermatogenesis refers to the process of producing sperm. Spermatogenesis begins at puberty, which is, again, in males, typically the young teenage years, 12, 13, 14, and it does not stop with age. So as men age, sometimes later in life, sperm production might decrease some because testosterone production might decrease some, but it does not stop. So in a normal, healthy male, sperm production continues throughout life. It does not stop. Typical production of sperm is about 500 million sperm per day. So 500 million sperm every single day. Notice these characteristics are completely the opposite of women, right? Women stop at middle age. The egg production stops at middle age. Women only release 1 egg per month, right? This is very different. We know that spermatogenesis occurs in the seminiferous tubules. We know that sperm are produced in seminiferous tubules. This happens specifically in the wall of the seminiferous tubule, and sperm production starts at the outermost layer of that seminiferous tubule wall. And as each step in sperm production occurs, we move closer and closer and closer in towards the lumen of the tubule. So like if we took a seminiferous tubule and we cut it so you could look at the wall, right? So you have the lumen in the middle, the open spaces here in the middle, and then you have the wall. Sperm production occurs out here in the outermost layer of the seminiferous tubule. And then remember, there are multiple steps in the cell division, right? The cell goes through meiosis 1 and meiosis 2. And then there's even more that happens after that to get to the final sperm. So each of these steps, as they occur, we move in closer and closer and closer towards the lumen until finally we have our sperm enter into the lumen of the tubule. Spermatogenesis, or sperm production, involves a series of steps. Remember, there's multiple cell divisions that occur and multiple different cell types. So every time the cell divides or changes, we give it a new name. Remember, this happens in the seminiferous tubules, and we're starting at the outside of the seminiferous tubule, and then every time the cell divides, it's just moving in towards the lumen or towards the center of the tubule. This all starts at the very outside of the tubule with a stem cell called a spermatogonia. So this stem cell or spermatogonia divides via mitosis. Remember, mitosis is just normal cell division. So this one stem cell divides via mitosis and it produces 2 daughter cells that are identical to the original cell. That means each of these daughter cells has 46 chromosomes, has the full set of chromosomes. So we started here at the outside of our seminiferous tubule. We have this one stem cell, and that one stem cell is going to divide to produce 2 daughter cells. So now we have 2 cells. One of those is going to stay there as the spermatogonium, right? Because we need to keep the stem cells out there. Otherwise, we wouldn't be able to continue making sperm. So that one cell stays out there as the spermatogonia. Again, it's going to divide. One will stay there, another one will continue. It'll divide again, one will stay there, one will continue. And that process happens all the time. But there always has to be one of the cells that remains out there as the stem cell. The second daughter cell that we made, though, is going to differentiate, which that just means like mature, kind of change. It's going to differentiate or mature into a primary spermatocyte. So we have our original cell on the outside. Moving in a little further, we have our primary spermatocyte. Now that primary spermatocyte undergoes meiosis, our meiotic cell division, meiosis, and forms two secondary spermatocytes. So now this primary spermatocyte divides and gives us two secondary spermatocytes. So now we have two cells. Okay, so remember we had our Seminiferous Tubule. We started with the Spermatogonia out here on the outside, our stem cell. That Spermatogonia divided by mitosis to produce two cells. One of them is going to stay there as the Spermatogonia. One of them moves on. Now we start our meiosis. We have meiosis 1 that occurs first, where that primary spermatocyte divides into two, and that gives us our two secondary spermatocytes. Okay, now these secondary spermatocytes have 46 chromosomes. Okay, so they have 46 chromosomes still. Now these secondary spermatocytes are going to undergo meiosis 2. That's our second meiosis cell division or meiotic cell division. So each of those divides. Now we have 4 what we call spermatids or immature gametes. Remember that this last cell division did not involve any duplication of DNA, right? So we had 46 chromosomes in each of these red secondary spermatocytes, and when they divided, they split up all of those chromosomes. So now each of these 4 spermatids in purple, each of those only has 23 chromosomes. Now, so we had our whole meiosis has occurred. We now have 4 daughter cells, but those 4 daughter cells, they're not actually sperm yet, right? They're immature. We have this last process that has to occur called spermiogenesis. Spermiogenesis, again, is the last step of spermatogenesis, right, or the last step of sperm production. In spermiogenesis, these spermatids become functional sperm cells or functional spermatozoa. Right now, before spermiogenesis occurs, these 4 spermatids are actually all connected to each other still. There was no actual division of the cytoplasm in meiosis 1 or meiosis 2. So we were dividing up our DNA and pulling it all apart, and we've got all of our cells here, but they're all connected to each other. So spermiogenesis is important because spermiogenesis is when we actually separate all of the cytoplasm and the cells differentiate into sperm, which there's a huge difference. Sperm look very, very different than the original Spermatogonia do. They look very different than most cells do. So there's a lot of changes that occur, like shed cytoplasm, right? The flagellum is sticking out. There's all of those changes are occurring as these spermatids differentiate into the sperm or spermatozoa. The spermatozoa, right, after they go through all of this, they're then in here, like right by the lumen or the open part of the tube. So the spermatozoa will then lose contact with the wall of the seminiferous tubule and enter into the lumen. So they'll go from the inner wall into the lumen, into this open space here in the middle. That's called spermiation. So I know all of these words are really confusing, but they all sound so similar. But if you kind of talk yourself through the process a few times, it's helpful. So you'll say, you know, we have mitosis first with the stem cell, then we go through a meiosis, right? The primary spermatocyte undergoes meiosis 1. Then the secondary spermatocyte undergoes meiosis 2. Then we have our spermatids. Our spermatids undergo spermiogenesis, and then they enter into the lumen via spermiation. Okay? After you do it like 100 times, it'll make sense. This is showing the process of spermiogenesis, specifically the differentiation that occurs, or the way that the spermatid changes into the spermatozoa, right, or the sperm. This process generally takes about 5 weeks, so it's not instantaneous. It takes a while for this process to occur. Here you have the spermatid, and this looks like a normal cell, right? A round cell, the big nucleus in the middle. That's it. Golgi apparatus, mitochondria, all the normal organelles. And then if you look here at the end, the spermatozoa looks very, very different. So let's talk about a couple of the specific changes that occur. One, it sheds a lot of the cytoplasm, right? Notice all this excess cytoplasm here starting to pinch off and getting shed. The resultant cell is much more sleek. It has to be. The sperm has to swim up the vaginal canal. It has to be able to fertilize an egg. It needs to be able to swim well, so that shape is important. It has to shed all that excess cytoplasm so it's not so big and bulky. If you look at the Golgi apparatus, notice how the Golgi apparatus kind of all merges together and fuses together and remains right at the top of the nucleus, and it becomes this acrosome. The acrosome is going to be important when it comes time for the sperm to fertilize the egg. The components of that acrosome are really important for fertilizing the egg, which we'll talk about later. Also notice the mitochondria. The mitochondria start out all kind of spread throughout the cell, right? That's normal. But as the sperm matures, the mitochondria end up lining up all along the microtubule base of the flagellum. This long whip-like tail, that's the flagellum. And that flagellum moves in like a corkscrew motion, and that's what allows the sperm to swim, right? To literally swim up the vagina and fertilize an egg. And that takes a lot of energy, right? It uses a lot of energy to constantly be swimming. So the mitochondria produce that energy. Makes sense that the mitochondria would be lined up all along this base, which is like where the motor is to help the sperm swim. This is an actual picture from a microscope of a seminiferous tubule. So this is real. This is what it actually looks like. Looking at this, this is the tube, right? So this is like the outside of the tubule. And then this is the open area on the inside. This is the lumen. Remember that this is all starting like out here in the outer wall of the tubule. And all of these steps kind of bring us in further and further towards the inside until we have the actual spermatozoa, the actual sperm, that are then going to enter into the lumen. Also notice these cells that are kind of surrounding the outside here. These are not sperm cells. Those cells that are on the outside, those are the interstitial cells or Leydig cells. You can see up here too, they're labeled. Those are the cells that, remember, produce androgens like testosterone to stimulate this process, Okay, they stimulate the spermatogenesis. You can see the different types of cells. Spermatogonia are the large stem cells on the outside. They divide and will have primary spermatocytes and secondary spermatocytes as we move in. Here, these are a couple dividing spermatocytes. The spermatids are the cells that they haven't shed the cytoplasm, they haven't differentiated yet into the sperm. They have half the chromosomes, right? They're haploid, they have 23 chromosomes, but they're not the right shape yet. So those are going to be like the large round cells that are here towards the center. Those are going to be spermatids. Then they go through spermiogenesis, and you can see all of these like really little thin cells. Those are the actual spermatozoa that are going to be ready to enter into the lumen and go on their way. We've talked about this a lot, but this is just one more picture. This one's drawn, but one more picture to show you this process. Here you can see interstitial cells, right, or Leydig cells that are around the outside surrounding the area. These again produce testosterone. In the seminiferous tubule, so you can see this like it's going around the outside of the tubule, and then the lumen is in the middle. Again, the process starts out here on the outside, and where we are going to have our spermatogonia, and the spermatogonia these are out here. So these are the spermatogonia on the very outside. They're going to divide to give us primary spermatocytes, which you see the next cell in. These are primary spermatocytes. Those divide to give us secondary spermatocytes, and then the secondary spermatocytes divide to give us our spermatids. These spermatids, remember when they first, like here, these are the spermatids when they're first formed, and you notice they're not the right shape, right? So they have to undergo spermiogenesis, where they differentiate into the right shape. So this is the initial one before they've completed the process, and then completing spermiogenesis, you see they actually look correct now. They're thin, they have the flagellum sticking off, and then they'll enter into the lumen and continue on. We haven't talked about nurse cells yet. Nurse cells are these large cells that kind of surround the sperm as they're going through this process, and they aid in the process. We will talk about them in a second. So the seminiferous tubules have various types of cells throughout them. Starting from the outside, progressing in towards the inside, remember we have essentially the sperm cells, just at various different stages of development. So the seminiferous tubules contain all different types of cells in them. There are all of the normal sperm cells, right, from the stem cells all the way to the spermatozoa. But there are also the large nurse cells like I just showed you. Those nurse cells, again, play a supportive role in the process of spermatogenesis. If you look at this picture over here, the nurse cells are what's shown in gray. So like all of these gray areas here in between are showing you nurse cells. All of the green, that's the dividing spermatocytes as they're making their way from the outer kind of capsule in towards the lumen of the tubule. Notice that again, all like surrounding them are nurse cells. The nurse cells are attached to the tubular capsule on the outside, and then they continue all the way in towards the lumen. They extend in all the way to the lumen between all the other types of cells. So they come in and they surround all these other cells and extend all the way into the lumen. The nurse cells have multiple different functions in supporting the spermatocytes. One, the nurse cells are joined by tight junctions. We've talked about tight junctions before, for example, like in the brain with the blood-brain barrier. We talked about the endothelial cells have tight junctions between them. Well, the nerve cells are also joined together by tight junctions, and that creates a really strong barrier, a really hard to permeate barrier between the blood and the testes. So this isolates the inner compartment or luminal compartment of the seminiferous tubules where we have this spermatogenesis occurring. The cells are kept in this protected, really tightly controlled or tightly regulated environment as a result of this blood-testes barrier. So these nerve cells produce the luminal fluid that fills this luminal compartment, and they can regulate the composition of the luminal fluid because of the tight junctions that they form. The blood-testes barrier allows for the luminal fluid to have a very different composition than the surrounding interstitial fluid. So just to kind of summarize, the nerve cells, remember, surround the dividing spermatocytes, and the nerve cells form tight junctions between themselves so that there's a really strong barrier between the surrounding fluid and the fluid inside the luminal compartment. The nerve cells control the fluid, the ions, the nutrients, anything, hormones, whatever. The nerve cells control what is able to get across them into that luminal compartment. So they secrete, whatever they secrete into that luminal compartment becomes the luminal fluid. So nerve cells form the blood-testis barrier, and nerve cells produce and control the luminal fluid. This keeps the testes and the surrounding dividing spermatocytes in a very tightly regulated environment that is optimal for spermatogenesis. So we said nerve cells kind of encourage mitosis and meiosis in the tubules, and they really support the dividing spermatocytes. Nerve cells are stimulated by follicle stimulating hormone. Follicle stimulating hormone, remember, comes from the anterior pituitary gland. If you remember back to endocrine, remember we have the hypothalamus, and then underneath it you have the infundibulum, and that connects to the pituitary gland. The anterior pituitary in the front, and then the posterior pituitary in the back. We are really worried about the anterior pituitary gland right now. So the anterior pituitary gland is controlled by the hypothalamus, and in this case, the hypothalamus releases GnRH, gonadotropin releasing hormone, and that gonadotropin releasing hormone goes to the anterior pituitary, and that tells the anterior pituitary to release FSH and also LH, which we'll talk about a little bit later. So FSH is coming from the anterior pituitary gland, and FSH makes its way down to the testes, and it stimulates these nerve cells. And again, then the nerve cells help to support the mitosis and meiosis. They're also stimulated by testosterone. And we talked about testosterone a little bit ago, but remember, testosterone is coming from the Leydig cells. Now, it's important to note that testosterone only stimulates the production of sperm when there is FSH secretion. So FSH has to be present in order for that testosterone to be stimulating sperm production. So we need FSH and testosterone. The nerve cells then promote the mitosis of the Spermatogonia. Okay, so cell division of the Spermatogonia, which were those initial stem cells. And they support the meiosis of the spermatocyte, right? So the cell division of the spermatocytes, which was meiosis, from our primary spermatocyte to our secondary spermatocyte to our spermatids. Nerve cells also support spermiogenesis, which, remember, is the transformation of the spermatid to the spermatozoa, which is actually pictured here. Remember, the spermatid is just like a normal round cell. The spermatozoa looks like our typical sperm, like a little tadpole. The process of, turning from that normal round cell into the spermatozoa is spermiogenesis, and that's really stimulated and supported by the nerve cells. The way that the nerve cells kind of help or support this process is they surround and enfold the spermatids, and they provide nutrients to the spermatids, as well as the chemical mediators or chemical stimuli that are needed for development. The cells don't just automatically differentiate into the way they're supposed to look at the end. They need stimuli to stimulate them, to tell them, hey, this is what you do now. This is what you do now. They need instructions. And the nerve cells provide those instructions to them. And then again, as well as providing nutrients that they need. Also, the nerve cells phagocytize the cytoplasm and the extra waste stuff that gets shed by the spermatids. Remember that the spermatids shed cytoplasm, right? They get rid of a lot of excess cytoplasm that they don't need. And we don't want that junk just kind of clogging up the testes, so the nerve cells phagocytize it. They eat it or engulf it to get rid of it so that the area surrounding the sperm is nice and clear. Nerve cells also secrete a hormone called inhibin, and this gives the nerve cell control over the rate of spermatogenesis or sperm production. Inhibin is a peptide hormone. It's a hormone that's secreted by nerve cells in response to chemical factors that are released by sperm. That sounds kind of complicated, but just what it means is the more sperm that there are, the more inhibin that the nerve cell will produce. So when there's a ton of sperm around, that will tell the nerve cell, hey, we've got a bunch of sperm here already. So then the nerve cell will release inhibin. Inhibin, again, is a hormone. So inhibin travels through the bloodstream, and it's released by the nerve cells, and it travels up to the anterior pituitary gland. And inhibin selectively decreases the production of FSH in the pituitary gland. So pituitary production of FSH is decreased. Now, remember, we said FSH stimulates spermatogenesis. So by decreasing FSH, inhibin decreases spermatogenesis. So inhibin gives nerve cells negative feedback control over spermatogenesis. Remember, negative feedback is like when something starts to happen, so you then stop it from happening, right? So when we start to produce a lot of sperm, the nerve cells make inhibin. Inhibin decreases FSH, and then we have decreased sperm production. We'll spend a little bit of time just looking at the sperm itself, the anatomy of the spermatozoa, which is really important to its function. The spermatozoa, the body of it, can be divided up into the head, neck, middle portion, and tail. The head is the large rounded portion. That's where the nucleus is, which the nucleus is important because remember, that's where the DNA is. That's where those 23 chromosomes are that are going to have to be able to be combined in with the egg so that the offspring has the full 46 chromosomes. So the nucleus with the DNA is there, right? That's what chromosomes are. And also the head has the acrosome. Remember when we looked at the development, when the spermatid turned into the spermatozoa, we said that the Golgi apparatus or Golgi complex all like kind of melts together and becomes this acrosome, right? So the acrosome is like this cap on top of the head there, so a cap-like structure. This is a membranous compartment, so like a membrane-bound storage chamber that contains enzymes that are essential to fertilization of the egg. And again, we said that it was made from fused little chambers from the Golgi apparatus. Now, this makes sense that the enzymes that are needed to fertilize the egg are present here at the tip of the head, because the spermatozoa is going to be swimming, right, kind of like a tadpole swimming, and this head region is what's going to approach the egg first. So if the sperm needs to fertilize the egg, it needs to penetrate into the egg, it makes sense that those enzymes that help the process are right here at the tip of the head, which is what's going to hit the egg, initially. The neck is just this very tiny little connector, right? Just like in us. We have this big round head and then this narrow neck, right? There's the big round head and then the short narrow neck that connects to the middle piece. The middle piece has the mitochondria spiraling around the microtubules. Microtubules are just like the base of the tail. So you have this tail or this flagellum, and then it's got it's made of microtubules. And the microtubules continue in to form like a little base in this middle piece. And then the mitochondria you can see kind of spiraling around those microtubules. We know that mitochondria produce ATP, right, which is just cellular energy. So they produce the energy or fuel that's necessary to move the tail or flagellum. So it propels the sperm so that it can swim. The tail region is this flagellum. The flagellum is only present in the spermatozoa. There are no other cells in a human body that have a flagellum besides the spermatozoa. It's a long, kind of thin, again, whip-like organelle that beats rhythmically. It moves in kind of a corkscrew type motion in order to propel the sperm from one place to another. The testes produce sperm that are physically mature. So like all of the structures are present, they're the right shape, they look the way that they're going to look at the end, they're anatomically correct, but they're not functionally mature, meaning they're not actually ready for fertilization yet. They're not actively swimming yet. Essentially, they're just like non-activated sperm. So the sperm have to functionally mature before they're ready to be released into the vagina. The spermatozoa that are released into the lumen of the seminiferous tubule, they're incapable of locomotion. Okay, again, that's like movement. So they're incapable of movement and fertilization. So we need for them to mature before they get released. And that long process of maturation happens throughout the rest of the reproductive tract in the male. The sperm move from the testes from the seminiferous tubules of the testes back into the epididymis, and they're moved, they don't swim yet. So there are cilia that are present lining the little efferent ductules, and those cilia beat rhythmically. They move like this to push the sperm back into the epididymis. And then the sperm start this process of maturation back as they go through the epididymis. And all of these other parts of the reproductive system, not the testes, but other areas, are responsible for the functional maturation of the sperm and nourishing the sperm, storing the sperm until they're needed, because they're not released continuously, right? They're released during sexual intercourse. So there are other parts of the male reproductive system that are responsible for the storage and maturation, and that's things like the epididymis, the Ductus Deferens, the ampulla of the Ductus Deferens, etc. So we'll start with the epididymis. The epididymis is the start of the male reproductive tract. We know that it's a really tightly coiled tube. It's almost 7 meters in length, so almost 23 feet long when you stretch it out, but it gets coiled about this big along the back of each of the testes. So it curls along the back border of each of the testes. So there's an epididymis on each side. The functions of the epididymis include: one, it monitors and adjusts the fluid that's produced by the seminiferous tubules, recycles damaged spermatozoa so that the components can be reused again to make new sperm, and then it stores and protects spermatozoa, helping to facilitate their functional maturation. Here you can see the epididymis curled along the back of the testis. So this blue kind of circle in front, that's the testis, right? When you look inside, you can see the seminiferous tubules here. And then you see these little, the little ducts, the efferent ductils, that are bringing the sperm from the rat testis into the epididymis. So the epididymis, again, this tightly coiled tube along the back here, can be broken up into the head, right? The head of the epididymis is this larger round portion at the top of each of the testes. That's where the sperm initially go. So that's like the beginning of the epididymis. And then you have the body of the epididymis kind of curling around the back. The tail of the epididymis is then this end portion right here, right after it kind of curves to go back up again. And then the epididymis, you'll see, empties into the Ductus Deferens. So the Ductus Deferens is then this tube that continues up here inside what we call the spermatic cord. The spermatozoa that leave the epididymis are mature, but they're still immobile. So as they leave the tail of the epididymis and go into the Ductus Deferens, they're now mature, they're functionally mature, but they haven't actually started swimming yet. So to become motile, or actively swimming and fully functional, the spermatozoa undergo a process called capacitation. Now, really, we don't want that to happen yet. We don't want them to start swimming yet because they're not ready to be ejaculated yet. They're still, pretty deep in the reproductive tract of the male. But once we're ready for them to start actively swimming, then they have to undergo capacitation. Again, there are generally 2 steps in capacitation. The sperm become motile when they're mixed with secretions of the seminal glands. So the seminal glands make some of the components of the ejaculate, the semen. Some of the components come from the seminal gland, some come from the prostate gland, and then of course we have the spermatozoa as well. So when the spermatozoa get mixed in with the secretions from the seminal gland, that is one step in their capacitation. And then they get mixed with all those secretions, and the semen gets released from the penis into the vagina. And when the spermatozoa are exposed to the female reproductive tract, that's the final step for capacitation. That's the final step for them to be actively swimming. The epididymis actually prevents this process intentionally. The epididymis secretes chemicals that prevent capacitation from occurring too soon, because the sperm are only able to swim so much for so long. So if they're going to be, in the Ductus Deferens for some time, and they're not going to be released into the vagina right away. So you don't want them swimming for no reason. They're going to use up all of their energy and their lifespan, and then they're not going to be alive once they get released into the vagina. So it's important that they don't start that process of capacitation until they're actually going to be released. Here you see the area where the spermatozoa are going to meet up with the secretions from the seminal gland. What you're looking at is a back view. You're looking at the back of the bladder. This large circular structure here is the bladder. These are the ureters. So this is where like urine would come into the bladder. We're worried about the reproductive structures though. So what we're worried about is the Ductus Deferens. These tubes right here, these purple tubes on the sides. This is the Ductus Deferens that is going to be bringing the spermatozoa up. So the Ductus Deferens comes down, and remember that meets up with the epididymis. So the sperm are produced in the testes, right, in the seminiferous tubules. Then they enter into the epididymis. Then they go from the epididymis into the Ductus Deferens. They come up the Ductus Deferens. Eventually they get here to the ampule or ampulla of the Ductus Deferens, and then you can see all these little like tubes kind of come together at this point here in the middle, right? We have the sperm coming in from the ampulla, from the Ductus Deferens. Notice a little bit wider out from that. Here I'm circling them in yellow. These are the seminal glands. They're making fluid that goes into the semen. That's going to get mixed in with the sperm. And then here surrounding, just right underneath the bladder, surrounding the urethra, we have the prostate. And the prostate gland is also going to be making fluid that gets mixed in to form the semen. All of that is going to get mixed together and released into the urethra, right? So then all of the semen, including the sperm and all the fluid, is going to get released through the urethra. Now, again, this process isn't quick. Even just the initial sperm production takes a long time, right? For the spermatid to become spermatozoa, that's like 5 weeks. And that's not even starting from the very beginning. So we have the initial cell divisions that occur. And then we have the process of, differentiation to become the nice sperm. That's like 5 weeks. Then we have the time that we spend in the epididymis. The sperm can spend months in the Ductus Deferens. So, this process coming here and then kind of getting stored here, that can take months. So the sperm are in the male body for quite a while before they get released. Where the Ductus Deferens merges with the seminal gland, we form something called the Ejaculatory Duct. Right, so you'll notice like the Ductus Deferens coming down and the seminal gland coming down, and then that forms this duct right here is called the Ejaculatory Duct. And that ejaculatory duct is going to go through and flow into the urethra. And then again, it's the urethra that actually brings the sperm and the other components of semen out of the penis. The accessory glands of the male reproductive system refer to all of the glands besides the testes. The testes produce the sperm, right? But there's other components that are present in semen, and those are produced by the accessory glands. So the accessory glands produce the components of semen besides the sperm, not the sperm. The secretion or semen, is a combination or a mixture of secretions from multiple different glands. Again, these include things like the seminal gland and the prostate glands, like we just saw in the last slide. Also, we'll talk about the bulbourethral glands as well. They were shown on the last picture. They're smaller little glands that are down by the urogenital diaphragm, like the pelvic floor. The semen is important because semen acts as a vehicle for sperm transport, and it also supports the sperm and helps the effectiveness of the sperm, if you will, helps to increase the chances of the sperm being able to fertilize the egg. Semen contains between 20 million and 100 million sperm per milliliter. So it's very, very dense in sperm in a healthy individual. and then seminal fluid. So it's a combination of the sperm itself and the seminal fluid, which is produced from the glands that we just mentioned, the bulbourethral glands, the seminal gland, the prostate gland. The seminal fluid, again, acts as a transportation medium for sperm. Sperm are microscopic little cells. We can't just eject the cells from the penis. They need to be ejected in something else, and that's seminal fluid. Again, seminal fluid isn't just water. Seminal fluid has very specific components in it that help to nourish the sperm and feed the sperm and make sure that the sperm is successful in fertilizing the egg. It has, the seminal fluid has high concentrations of fructose. Fructose is a sugar, right, a carbohydrate, and it's easily metabolized by the spermatozoa. Remember that the sperm have all of those mitochondria, and they need to make ATP in order for the flagellum to move so they can swim. So all of the fructose that's present in the semen, it's like a bunch of nutrients that's packed in right there, ready for the sperm to be able to use to make ATP in order to swim. There are also prostaglandins in semen. The prostaglandins actually work on the female. So the semen gets released into the female vagina, and the prostaglandins stimulate smooth muscle contractions, and that helps to spread the sperm out. You don't want the seminal fluid to just get released into the vagina and then to drip out and leave, which we'll talk about more in just a second with Seminogelin. You want it to be dispersed throughout the female reproductive tract, and causing contractions in the female reproductive tract helps that happen. Seminogelin is also important for that. Seminogelin is a protein that forms a temporary clot in the semen, in the fluid, and that helps to keep the semen in the vagina. This is secreted as proseminogelin, which gets activated by enzymes from the prostate. You can think of this kind of like a normal blood clot. Remember the blood clot, it starts as fibrinogen, and then that gets converted into fibrin, and that forms a clot. Proseminogelin is soluble. It's soluble, so it dissolves in the fluid. But once that proseminogelin gets mixed with fluids from the prostate gland, it gets converted into seminogelin, which seminogelin is insoluble. So it forms a clot-like structure, right? It kind of clots up the seminal fluid. So seminal fluid is first liquid. It can be released or ejaculated from the penis, but once it gets released into the vagina, it'll thicken up. And if it's a thicker fluid, it's more likely to stay in the vagina, especially with the smooth muscle contractions that are occurring, and less likely to be to drip out, essentially to be released out of the vagina. We talked a bit about sperm motility before. We said that capacitation is the process that happens in order for the sperm to become motile, to actually start actively swimming. And we said that that doesn't happen until the sperm are in the female reproductive system, so until they're actually in the vagina. The reason for that is that sperm motility requires an alkaline pH. It requires a basic pH, so a pH greater than 7. The spermatic ducts are acidic, so the sperm are not motile in the spermatic ducts. Now, the vagina is also typically acidic, so that's problematic. The vagina needs to be acidic for vaginal health, for the flora that naturally live there to be healthy. However, upon ejaculation, there are alkaline secretions from the prostate gland that create a more alkaline environment in the vagina, and that activates the sperm. And so that alkaline secretions from the prostate, those are important so that the environment in the vagina is the right pH for the sperm to be motile and for the sperm to actively swim. We said that sperm motility requires energy, right? The sperm need ATP. That energy source is provided by the fructose from the seminal vesicles. Mixing sperm with seminal fluid initiates that first step in capacitation, okay, when we have the appropriate pH present. The spermatozoa begin beating their flagella. The flagella starts moving and they become highly motile, and they swim up the vagina and uterus, and inside the uterus is where they should meet with the ovum or egg that they can then fertilize. We'll take a second to talk about the hormones of the male reproductive system. We have mentioned them a little bit. We talked about GnRH and FSH and testosterone, but we'll just kind of sum them up again right now. So beginning at puberty, the hypothalamus starts to release GnRH, or gonadotropin releasing hormone, and it releases it in pulses. Okay, so it just starts, every hour, hour and a half, it releases a pulse of GnRH. We know that GnRH from the hypothalamus stimulates the anterior pituitary gland, and it stimulates the anterior pituitary gland to release FSH, follicle stimulating hormone, and LH, or luteinizing hormone. The FSH that's released from the anterior pituitary gland and testosterone, okay, so the combination of those two things, FSH and testosterone, target the nurse cells that surround the developing sperm in the seminiferous tubules, and FSH and testosterone stimulate them to promote spermatogenesis and spermiogenesis. So essentially you could say FSH and testosterone together stimulate sperm production. Now note that testosterone has to be with FSH in order to stimulate sperm production. If there's no FSH, if it's only testosterone, then testosterone won't stimulate sperm production. It'll have other effects though. Then the testosterone will just have its other effects in other areas of the body. Like remember, like the secondary male sex characteristics, like voice, hair growth, face, that sort of thing. But so testosterone can have other effects on the body. In order to stimulate sperm production, that testosterone has to be with the FSH. Luteinizing hormone, right, LH, targets the Leydig cells or interstitial cells around the testes. And those interstitial cells will then secrete androgens, right? So testosterone and other androgens like dihydrotestosterone. Okay, so that testosterone, remember, can be important for stimulating sperm production, but the testosterone also has other effects, right? It also has other systemic effects, which we'll talk about in a second. Spermatogenesis, so the production of sperm, is controlled by negative feedback. There's negative feedback control that can decrease the production of sperm when there's already plenty being made. In general, spermatogenesis or sperm production is regulated by GnRH, FSH, and inhibin. GnRH causes the release of FSH, right? And we know that this FSH stimulates spermatogenesis. As spermatogenesis accelerates, we have more and more sperm around. And remember, those sperm stimulate the secretion of inhibin from the nurse cells. Inhibin then goes up and inhibits the release of FSH. Now that's selectively. Inhibin inhibits the release of FSH. It does not inhibit the release of LH though. Right, so we have GnRH from the hypothalamus. That goes to the anterior pituitary and causes FSH and LH. FSH causes sperm production. LH causes testosterone release. If we have a ton of sperm production, right, if we've got a whole lot of sperm being produced, then that means we'll release inhibin. And inhibin is going to come up and inhibit the release of FSH. Okay, that decreases sperm production. So that's negative feedback, right? Inhibin then slows sperm production, but it does not affect the production of testosterone because look, we still have LH, right? Luteinizing hormone is still being released. So that means we still produce testosterone. That's important because testosterone is needed to maintain the other male sex characteristics. When we have plenty of sperm available, we don't want to then stop the other male secondary characteristics. We need those to continue. So this allows us to have separate control of sperm production and testosterone production. Testosterone. Again, testosterone is one of the most important androgens. Testosterone stimulates sperm production, right, as long as FSH is present, but it also has other effects throughout the body. It stimulates libido or sex drive and the related behaviors. It stimulates metabolism, establishes the secondary male sex characteristics like the distribution of facial hair, increased muscle mass and body size, characteristic adipose tissue deposits. It maintains the accessory glands and organs of the male reproductive tract, like the prostate gland, for example. Really high testosterone will inhibit GnRH. So when testosterone is really, really high, there's negative feedback. It'll decrease GnRH, which then decreases FSH and LH, which we know luteinizing hormone stimulates testosterone, so that will then decrease testosterone. This is just showing you the kind of hormones that are present in the male reproductive tract. This is the hypothalamus up here. This is the anterior pituitary gland here. So you can see GnRH coming from the hypothalamus, and it releases GnRH at pulses, right? Starting at puberty and continuing through the rest of life, there's just a periodic pulse of GnRH. That goes to the anterior pituitary gland and says release FSH and release LH. LH targets the interstitial cells of the testes and says to produce testosterone. FSH targets the nurse cells of the seminiferous tubules and causes sperm production. Remember, when there's a whole lot of sperm present, that causes the release of inhibin from nurse cells. And you can see inhibin's action here. Inhibin is coming and stopping the production of FSH. There's a question here if some levels of inhibin might interfere with GnRH, but that's it's not nearly to the degree of the FSH. So we'll just, for our purposes, know that inhibin inhibits the secretion of FSH, and that's it. Really high testosterone levels can inhibit the release of GnRH, which would then decrease the release of all the rest of these, right? It would decrease FSH, LH, and testosterone. This is just the bottom of that chart. So it was just showing you like LH stimulating interstitial cells to produce testosterone, and then here we have the effects of testosterone. This is FSH stimulating nurse cells, and that stimulates or facilitates spermatogenesis and spermiogenesis. Also, if we have an increased sperm, we have an increase in inhibin, and up on the other slide you saw that inhibin then inhibits FSH release. This is just showing us male versus female gamete production. We'll talk about the female side over here in the next lecture. This is the male side that we've looked at already. And before this, there was a mitosis that occurred. We had our spermatogonia, our stem cell, and our stem cell divided via mitosis to give us one spermatogonia that's going to stay up here and one primary spermatocyte. Then we have our meiosis. This is meiosis 1, where our primary spermatocyte divides into two secondary spermatocytes. Each of them are diploid. They each have 46 chromosomes. Our secondary spermatocytes divide to give us 4 spermatids. These are each haploid. They each have 23 chromosomes. But we end up with 4 spermatids, 4 sperm, 4 usable cells when the male gamete is produced. In the next lecture, we'll look at the female gametes, but you guys will see that in the female, we only end up with 1 mature ovum, 1 mature egg. The rest of these are what we call polar bodies. They're just not viable. Essentially, in the female, we put all of our resources into that 1 egg, and the other 3 just kind of die. Okay? We have to put everything into that 1 ovum to make sure it's really good and really quality. Okay? All right, that's it. I'll see you guys next time for the female reproductive system. Thanks for watching.